Let's begin lecture 4, which is eukaryotic gene regulation. We're going to talk about eukaryotic transcriptional activation and eukaryotic transcriptional repression. Do your readings as per usual. Now we want to start off this lecture with a reminder slide from a couple of lectures ago. So you've seen this slide before, there's nothing new here. Just as a reminder, gene expression in both prokaryotes and eukaryotes is regulated by gene regulatory proteins, these transcription factors, right? So gene regulatory proteins, these are transcription factors. And these proteins bind specifically to regulatory regions of DNA, also known as cis elements. Now, if we look at these transcription factors, these proteins, these specific gene regulatory proteins, we can actually divide them up into either activators or repressors. So you have gene regulatory proteins that can turn genes on, positive regulators, those are our activators, and then the ones that turn genes off, that repress transcription, there are repressors. Now we're going to really address this concept, these transcription factors, these activators and repressors in this lecture in eukaryotes. We want to start off with a very quick review. So hopefully you recall from your prerequisite courses that DNA is transcribed into RNA by the enzyme RNA polymerase. I'll go through this figure really quickly just as a very quick review. Remember, you have your RNA polymerase here in blue. Here is your DNA double helix. Here they show you the direction of transcription. So you're going to be reading the template DNA from 3' to 5'. Here are your ribonucleoside triphosphates. You're going to have an uptake channel on the RNA polymerase. And at the active site, you're going to cut off 2 of the phosphates and add on the next phosphate sugar base. So you're adding on to the 3' end of RNA. Here's the 5' end of the RNA. You are making the RNA from 5' to 3', as in you're adding on to the 3' end. You will have a short region that is a DNA-RNA hybrid, and then they'll separate and you have an exit channel where the RNA starts to leave. And again, that is a very quick review of transcription. This should definitely not be new, but obviously from your DNA template strand, you'll transcribe and make your RNA. Now, also as a review, and again, this should not be new if you're in second year biology here at U of T, cells produce several types of RNA. So here is our standard table. You don't have to memorize this complete table now, but we're going to talk about some of the other kinds of RNAs. Definitely you should know the first 3, right? So if you're in this class, definitely you should know mRNA is the messenger RNA, it codes for proteins. rRNAs, ribosomal RNAs, tRNAs, transfer RNAs. And you probably should know a lot more than just those 3 lines. And definitely some of these other RNAs we'll talk about as we go. Do recall there are some RNAs that don't help you make protein, right? Like microRNAs, miRNAs, siRNAs. These 2 kinds of RNAs actually do the opposite. They block translation. They actually stop you from making a particular protein. Hopefully you also recall that different RNAs are transcribed by different RNA polymerases in eukaryotes. That's different than prokaryotes. Prokaryotes have a single type of RNA polymerase. In eukaryotes, there are typically 3 different kinds of RNA polymerase. Now, I wouldn't memorize this entire table, but definitely you should know that RNA polymerase 2 does the protein coding genes. And definitely you should know that there are 3 types of RNA polymerase in eukaryotes. Of the different types of RNA we see in this particular table, the first 3 will be in both prokaryotes and eukaryotes, and then the other ones here will be only in eukaryotes. Prokaryotes may have some other ones, but we'll address those as they come up. Transcription initiation in eukaryotes requires many proteins, and these proteins are usually called general transcription factors. These general transcription factors help position RNA polymerase at eukaryotic promoters. Eukaryotic promoters often, sometimes contain the title box, definitely not always. Here's a table showing various general transcription factors. You can take a look at how they're named. So for example, you have TF as in transcription factor 2, so for RNA polymerase 2, and then subunit D. You'll have other ones all named using a similar pattern, so TF as in transcription factor 2 for RNA polymerase 2, B, subunit B, etc. Now you don't have to memorize this entire table. A couple of key ones include TF2D, because that contains the TBP subunit. TBP as in TATA binding protein, so it recognizes the TATA box, and it also has other transcription activation factor subunits, TAF subunits. This is how it typically functions. This should be a good review again of first year biology. If you don't have the appropriate prerequisites or you don't remember it, remember it's your responsibility to check. But as a quick reminder, a lot of your eukaryotic promoters will have the TATA box before the start site of transcription. TBP, TATA binding protein, a subunit of TF2D, so you know it's RNA polymerase 2 in this case, is going to bind to the TATA box. Other general transcription factors will bind. Another key one is TF2H. H, you can think of helicase, so it's going to help you separate the two strands. RNA polymerase 2 gets on here. And remember, RNA polymerase 2 has a C-terminal domain. The C-terminal domain tail gets phosphorylated by TF2H, and then transcription can begin. So if you think about your general transcription factors, these are required by nearly all promoters used by RNA polymerase 2. Now, a key bit of terminology here that can be a little bit confusing is that we use the term general transcription factors for these kinds of proteins, TF and then a number and subunit, right? Like TF2B, TF2D, these are general transcription factors, as in these are proteins you need for any kind of transcription. Now, in addition to these general transcription factors, we also have other transcription factors, and these would be the activators or the repressors that then activate it more or repress it. Okay, so separately, you also have these other transcription factors. Again, to review your eukaryotic transcription, RNA polymerase 2 transcribes the protein coding genes. It requires 5 general transcription factors, your TF2D, TF2B, TF2F, TF2E, and TF2H. Prokaryotes only need one, they just need the sigma factor. Eukaryotic genomes lack operons. Eukaryotic DNA is packaged into chromatin, which provides an additional mode of regulation. Eukaryotic transcriptional activation requires many gene regulatory proteins, as in many transcription factors. And here's an example of an overall picture. You can see the line here representing the DNA. Now the DNA is not broken here, it's just really long. You can see that you can have this particular region, say, being the promoter, and then in red is maybe the transcriptional start site. You can see at the promoter, you have a lot of general transcription factors, the TF2Ds, etc. You have the RNA polymerase on here. You can see the C-terminal domain tail. Now, really far away, you might have one of these other transcriptional factors, maybe an activator. Activator might bind to another DNA sequence, which will then activate the protein, causing it to then, say, interact with all these other proteins. Maybe through a mediator, it can interact with all these other proteins. And you might have additional enzymes like a histone modifying enzyme or chromatin remodeling complex to deal with the packaging of DNA. Now, your cells don't have to use this type of a protein, but it can. There are proteins like these mediators that act as an intermediate between the regulatory proteins and the RNA polymerase. So here we see the same diagram, just a bit larger. And if you think about it, how is this one activator protein supposed to really help activate gene transcription in eukaryotes when you have all these other proteins being involved? Well, one option is to bind to a mediator and then cause the mediator to then activate all these other things. Eukaryotic gene expression is controlled by many regulatory proteins. There's about 2,000 encoded by the human genome. So think about your genome, maybe 20,000 to 30,000 protein encoding genes. 2,000 of that is gene regulatory proteins, includes activators and repressors. That's a huge contribution. That's huge importance by these proteins. Gene regulatory proteins can act over very large distances, sometimes greater than 10,000 base pairs away. And as we said before, one mechanism is this DNA looping. Let's take a look at our schematic one more time. This shows us our eukaryotic gene in a little bit more detail, as in here's the gene region. Imagine this is the start of transcription. And of course, you have a promoter region. For eukaryotes, you might have a TATA box, you might not, depends on the promoter. And you have all these general transcription factors like the TF2D, TF2H, all that stuff. Here's your RNA polymerase II. And further back, notice you can have these gene control regions in green, these regulatory elements, these DNA sequences. They can be pretty far back, pretty far back. Don't forget, in eukaryotes, they can be after the gene too. They can be in introns. So they can be kind of all over the place. And there's gene regulatory proteins that will bind to them, right? Specific activators, specific repressors that will bind to those specific regulatory sequences. So notice the gene control region for gene X can be quite comprehensive, can include all sorts of different sequences far before, far after, in the introns possibly, and includes the promoter too. A lot of regions to control gene expression. And how do you input all this information into what you should do at transcription? Well, you can have quite extensive DNA looping. So imagine all these different regions. You may have, say, activators, you may have repressors. And one thing the mediator often does is it sums up all that input, right? So it says, oh, I got 3 activators here. Oh, we're really going to activate gene transcription. Or maybe I have some activators here and maybe 1 repressor. Well, maybe then we'll activate it, but not as much as we would if it was all activators, right? So the mediator can take up all the different kinds of information here and then really influence the gene transcription. The eukaryotic gene regulatory proteins often function as protein complexes on DNA. Now, just in a solution, you might have a number of these proteins floating around, but they may assemble on the DNA. So there's a couple of examples here just as schematics. So let's say this is your DNA. Let's say there's a blue sequence and a green sequence. And remember, your gene regulatory proteins are going to be specific, right? So a blue protein binds to this blue sequence, green protein binds to this green sequence, and they may recruit another protein, and together this complex will activate transcription. Here's another option, maybe in green, you bind to this green sequence, red, you bind to this red sequence, they recruit another protein, and together again, as a protein complex, notice in this case, you repress transcription. Now, if you look at this protein right here, notice it definitely affects transcription, but this one right here actually doesn't bind to DNA on its own. Same thing over here. This protein over here definitely can affect transcription, but it doesn't bind to DNA on its own. And so co-activators and co-repressors assemble on DNA-bound gene regulatory proteins and do not directly bind to DNA. Now, what we need to do right now is to start a little mini section really examining activator proteins. If you didn't understand the first section of a lecture, definitely go back and review. As well, always make sure to take a look at the textbook, but understand that stuff before you start this section. We're going to start looking at activator proteins. Our activator proteins definitely have a modular design in that they typically have 2 different domains, 2 different sections. They're going to have a DNA binding domain and an activation domain. So their specific DNA binding domain is going to help them recognize a specific DNA sequence, and your different activator proteins will recognize different DNA sequences, so they'll have different DNA binding domains. And then they'll also have this activation domain, which they use to accelerate the rate of transcription, to increase the frequency of transcription. So let's look at an example up here. Here is our Gal4 protein. So Gal4 protein, that's a typical eukaryotic activator protein. And so if it's a typical eukaryotic activator protein, you know it's got a DNA binding domain shown for you down here. It's got a activation domain shown for you up here. Now what happens is the Gal4 protein is going to recognize a specific sequence, right? Because it's got a specific DNA binding domain. And when it has recognized a specific sequence, then its activation domain will be activated. Then it can help activate transcription, so activating other proteins, etc. And then you'll activate, say, as an example, the factors at the TATA box, and you get gene transcription, right? So again, you need your DNA binding domain recognizing a specific sequence. When the protein is on the DNA, then the activation domain's activated. Then you get activation of transcription. Now, what happens in nature as well, what you can do in the lab is that very often there are mutations, there are kind of swaps where the DNA binding domains and activation domains kind of get mixed up a little bit. So over here, for example, you still have the green Gal4 activation domain, but notice now you have a different color down here. You have a different DNA binding domain. This is, in fact, the LEX-A DNA binding domain. So if you have a different DNA binding domain on this protein, well, you're not going to recognize the Gal4 sequence anymore, right? So you're not going to get transcription here. The DNA binding domain's different. You're recognizing a different sequence. Now, instead, what's going to happen with this kind of chimeric protein is that now you're going to recognize the DNA sequence for LEX-A. Whatever the LEX-A protein normally recognizes, that's the DNA sequence now you bind to. And when this protein's bound to this particular sequence, well, again, the activation domain, I know it's a Gal4 activation domain, but it's still an activation domain. Now it's going to activate transcription at this particular gene, right? So at this particular region where the recognition sequence is. So again, if you, say in evolution, get a swap, or if you in the lab do a swap, well, you still need the DNA binding domain to recognize a specific DNA sequence. And then, depending on what domain you put here, if it's an activation domain, then you get activation. So that is how your eukaryotic activator proteins work. They definitely have this modular design. Two important domains. So the activator proteins, yes, they have that modular design. They have the DNA binding domain and activation domain, but how do they activate transcription? Well, what we've already seen is that they can attract, they can position, they can modify things like the general transcription factors. They can affect the mediator. They can actually directly affect RNA polymerase II. And they can actually do this either directly or indirectly, in that they can directly act on these components and indirectly activate transcription by modifying chromatin structure. Now, over the next couple of slides, if you get confused, refer back to this slide. We're going to talk about several mechanisms that go through 1 and 2, right? So again, 1's direct mechanisms by these activator proteins, and 2 is activator proteins indirectly modifying chromatin structure. Now, these are not official numbers, so don't think there's officially a type 1 mechanism or type 2 mechanism. No. This is just an outline for our particular lecture. Okay, so let's start with number 1, how the activator proteins directly act on these components. And this is really the concept and the figures we've been talking about before. Activator proteins can bind directly to transcriptional machinery or the mediator and attract them to promoters, like, say, how prokaryotic activators do it. So again, imagine you have these gene regulatory proteins on the various regulatory sequences. You can use extensive DNA looping. You can act through a mediator. You can activate these components directly. For the second method, remember, activator proteins can act indirectly. And how they can do this is to alter chromatin structure. First, we have to make sure you remember your nucleosomes. So if you recall, nucleosomes are the basic structure of eukaryotic chromatin. We see DNA wound around a histone octamer. So there are different histone proteins. And the histone octamer consists of H2A, H2B, H3, and H4. And it is each of these proteins times 2. So if you look here, it's not just H4 times 2. You have 2 of H2A, 2 of H2B, 2 of H3, 2 of H4, and that makes up 8 proteins in the nucleosome core particle. So this is how it looks on a figure. You can see in red, this is your DNA. So imagine that's your double-stranded DNA. You can see it wraps around these proteins. In the middle here, you'd have that histone octamer shown for you in yellow. 8 proteins. They don't show you, but on the outside, you have one of H1, one protein of H1, and the H1 is kind of like a paper clip, clipping it on. You have this linker DNA, this DNA in between the protein beads, and of course, what you call the section in the middle are core histones, those 8 proteins of the nucleosome. And a nucleosome includes about 200 nucleotide pairs of DNA. This is what we call a beads on a string form of chromatin, as in the DNA looks like string and the proteins look like beads. Typically what we'd see is the DNA wrapped around the bead about 1.67 times, so it goes around once and another 2/3 of a time. There's about 147 base pairs that are wrapped around the core histones. The linker can vary between 10 to 80 base pairs. All these numbers will vary slightly though, depending on the particular region. Nucleosomes will pack as compact chromatin fibers. So what then happens is you have an additional level of packing, and there are currently two models as to how this beads on a string form of chromatin packs even more tightly. In the zigzag model, you can see that it looks essentially like a zigzag going up and down here. And for the solenoid model, this is essentially a solenoid shape. You can Google what a solenoid shape looks like, but that's essentially it. There's actually evidence for both of these models. However, in either case, transcriptional machinery cannot assemble on promoters with this tightly packaged chromatin. Let's say it's the zigzag model. Well, if this is a TATA box in here or a gene coding region, you can't have proteins get in there. You can't have general transcription factors get in there, or you can't have RNA polymerase get in there. It just doesn't fit. Same thing with a solenoid model. You can't get your proteins in there. So what happens is activator proteins can alter this chromatin structure and increase promoter accessibility. And there's a couple of ways they can do this. We'll go over the 4 major ways activator proteins can alter chromatin in great detail, but let's just look at this slide for an overview. And again, these are not official numbers. They're just kind of an outline for your slide to help you keep organized. But there are 4 standard ways shown for you here. Again, in red, let's imagine that's your DNA wrapped around those nucleosome core particles, and imagine the DNA is very compact. Let's say this is a TATA box or some other promoter region. You really can't get access to that, right? It's really buried deep inside, and you're not going to be able to transcribe this gene, right? The transcriptional area is very compact. So what happens is a gene regulatory protein can bind. Notice this gene regulatory protein is drawn for you quite well. You do see a DNA binding domain, binding to the DNA, and you do see an activation domain. And the activation domain can function in, say, these 4 indirect ways, right? So the first 3 actually all involve the chromatin remodeling complex shown for you here. So in method number 1, notice it calls in the chromatin remodeling complex, and it remodels the nucleosomes. It kind of rolls them around a bit, gives you a little bit more space. In method number 2, you get the chromatin remodeling complex. In addition, you get a histone chaperone, and the histone chaperone can actually remove the histones, right? So you remove the nucleosome core particles, you remove the histones. Definitely now it's much more accessible. There's a lot of room here. Third method is that, again, you use a chromatin remodeling complex. You again use histone chaperones, but these histone chaperones might be slightly different in that they might exchange some of the units here, exchange some of the histone proteins here, and maybe you now have a more loosely binding histone set in there. Fourth method does not really involve a chromatin remodeling complex. We'll talk about how there might be some overlap, but the fourth method involves a histone modifying enzyme where you actually modify the histones directly. So again, this is the overview. Let's go through the major ways one at a time. Nucleosome structure can be altered by chromatin remodeling complexes in an ATP-dependent manner to increase promoter accessibility. Our first major way was nucleosome sliding. So remember, this is your DNA, and you can have one of these ATP-dependent chromatin remodeling complexes come in. They can use ATP, and they can essentially catalyze nucleosome sliding, kind of like rolling the proteins here along. The analogy that I often give, which you should definitely not do, is something like toilet papering a house. You have your DNA as the toilet paper, your core proteins as the roll, and you're kind of rolling it along a little bit. Major way 2 and 3 involved nucleosome removal and nucleosome exchange, which requires cooperation with histone chaperones. Now in the figure, they don't quite show it to you in the exact order, but it's definitely all here in that you have, again, an ATP-dependent chromatin remodeling complex. One option is to also include a histone chaperone. Notice there's going to be ATP involved in the process. Definitely this is an ATP-dependent chromatin remodeling complex. The histone chaperones may exchange the dimers or exchange the proteins in the middle. And you might have something here that's a bit more loosely binding. Typically for our nucleosome core, you know there's 2 of H2A, 2 of H2B, 2 of H3, 2 of H4. Well, what happens is you can actually swap out H2A and H2B for what's called H2AZ and H2B. And definitely that can change the constituent proteins in here. That can definitely make it looser binding. But how that works is a little bit more complicated than we'll talk about in this course, but the main concept's there. Another option is, of course, to use a histone chaperone to remove the histones, and definitely now it's a lot more loose. Now, this next step is also an option, although you don't have to do it. So yeah, over here is loose. You can stop there, right? You can definitely have transcription here. Another option that is sometimes seen is that with a histone chaperone, you put in a different octamer. And very often this different octamer may have different functions, helping other proteins to bind as well. But this is just an additional option. You don't have to do this. Let's do a quick review of what we talked about so far in this little mini section where we talk about activator proteins in some detail. We're talking about how activator proteins activate transcription. Remember, they're going to attract, position, and modify your general transcription factors, the mediator, RNA polymerase II. We talked about how they can do this either directly by directly acting on these components, and more recently we just started talking about how they indirectly modify chromatin structure. And for this indirect method, remember there are 4 major ways. The first 3 involve this chromatin remodeling complex where you move stuff around. Maybe you also use a histone chaperone. You can remove the histones. You can change the subunits. And the 4th method is this histone modifying enzyme method. And that actually produces signals for chromatin remodeling. For our 4th major way, histone modifying enzymes produce specific patterns of histone modifications. Now, when you modify the histones, the modifications you put in, that's called a histone code. There are different ways you can modify histones shown for you here. You can add a phosphate group. The enzyme that adds a phosphate group is called a kinase. You can add an acetyl group. That's called acetylation. The enzyme that does that is called an acetyltransferase. You can add a methyl group. That's called methylation. The enzyme that does that is a methyltransferase. Importantly, there are kinases, acetyltransferases, methyltransferases that act on other proteins and act on other proteins elsewhere. But here we're talking about histone modifications, modifying the histones. The histone modifications occur on specific amino acids of the histone tail. Here you can see your nucleosome core particle. So of course the DNA is wrapped around it. Now remember, for a nucleosome core particle in here, we have 2 of H2A, 2 of H2B, 2 of H3, 2 of H4. Now interestingly, what happens is their tails actually stick out. And typically we consider modifications on N-terminal tails. So you can imagine the N-terminus, there's a portion that sticks out. N-terminus of this one, there's a portion that sticks out. N-terminus of this one, there's a portion that sticks out. More recently, people have been studying also what happens on C-terminal tails that stick out. So those definitely can be modified too. But yeah, N-terminal modifications can occur. So again, what we see are specific modifications to the histone tails. Again, N-terminal tail and C-terminal tail, they can be modified. And the modifications are done by histone modifying enzymes. Those enzymes we just talked about, the kinases, methyltransferases, acetyltransferases. And if you modify the histone tails, if you're one of these enzymes that changes the tail, well, we call you a histone writer, like you write on the tail. Here is one example. You don't have to memorize this example. This just shows you one example. This shows you histone H3. So you can see histone H3, the tail sticking out here, as well the other histone H3, the other tail sticking out here. And again, N-terminal tail shown for you here, but could be C-terminus. Now we can count the amino acids, right? From the N-terminus, it's 1, 2, 3, and all the way to the 9th amino acid. And the 9th amino acid in this case happens to be a K, right? Happens to be a lysine. It might be methylated. You can also have the 4th amino acid and the 9th amino acid modified. Say this one's methylated, this one's acetylated. Or maybe the 10th amino acid, which is actually a serine, gets phosphorylated. 14th one gets acetylated. There are all sorts of different modifications that can occur on the tails. Again, all sorts of different histone modifying enzymes known as writers. Now, if there are writers, as in they write a histone code, there's going to be histone code readers. And so reader proteins can recognize specific modifications and provide meaning to the code. Here's an example. Again, you do not have to memorize this example. It's just important for you to understand the concept that maybe a writer comes in and then modifies histone H3, modifies the tail, adds a methyl group onto the lysine. Then a reader is going to come in and read this code and say, oh, that's going to be heterochromatin formation. We're going to silence this gene. Or maybe with a different set of modifications in histone H3, readers will come in, recognize this code, and say, oh, that's gene expression. We're going to do something there to change it. Similarly, down here, a different code would also cause gene expression, and another code might cause silencing. And this is just for histone H3, but it's going to be a combination of modifications on all the different histone tails that really cause a particular output. Let's do another quick review because that was pretty complicated. So remember, we're talking about activator proteins, we're talking about this indirect method of activating transcription, and we said there are 4 major ways. We talked about the first 3 in great detail, and kind of the one we just addressed was number 4, these histone modifying enzymes and how they provide a signal for chromatin remodeling. So remember, there are specific histone modifying enzymes. These histone modifying enzymes, they're writers. They're going to write on the histone tail. They're essentially going to write a histone code. Now, there are many different kinds of enzymes that can function as histone modifying enzymes that can function as writers. You have kinases that phosphorylate things. You have acetyltransferases that add an acetyl group. You have methyltransferases that add a methyl group. And where do you modify the histones? Well, again, it's on the histone tails. You can modify the N-terminal tails, you can modify the C-terminal tails, and depending on how you modify them, depending on how you write with them, while you produce a different code, it's going to be read differently. So there's going to be specific code reader proteins that recognize the modifications that those enzymes have done, and they produce a particular meaning. So that's the overall concept. But let's talk about a specific example. You should know this example as well. It's going to help you understand the general concept as well. We're looking at an example of transcriptional regulation using the histone code, and the specific example is for the human interferon gene promoter. The interferon gene is involved in inflammation and immunity, but that's not really the discussion here. The discussion here is the regulation. So as an example, you can see you have the DNA double helix over here. You can see the TATA boxes here as well. There's another cis element here, another DNA sequence here. Notice the DNA is wrapped around the histone core. They don't show you all the proteins, but of course you'd have 2 of H2A, 2 of H2B, 2 of H3, 2 of H4 with the tail sticking out. And so you can imagine you can't really access the gene here. You can't really transcribe. It's really compact. They show you a number of different steps. Don't forget, you cannot skip steps. One step has to occur before you get the next step, okay? So you cannot skip steps. So in step number 1, it says an activator protein binds to chromatin DNA and attracts a histone acetyltransferase. Notice the short form for histone acetyltransferase is a HAT. So exactly as is described, you have a gene activator protein shown for you here, and this one's drawn for you quite well. Notice it has a DNA binding domain to recognize a specific DNA sequence, has an activation domain. And how this particular activation domain works is it calls in a histone acetyltransferase in blue here. So it calls in a HAT. In step number 2, the HA, as in the histone acetyltransferase. So you can write HAT or HA, that's fine. But the histone acetyltransferase acetylates lysine 9 of histone H3 and lysine 8 of histone H4. So the histone acetyltransferase puts the acetyl group, shown for you as this blue square, dark blue square. Notice the symbols here. Know the symbols. H3K9, histone 3 lysine 9. You add the acetyl group. You add it to the other histone 3 lysine 9, right? The other tail over here. You add it to the histone 4 lysine 8, H4K8, you add it to the other one. So step 2 is now complete. You can proceed to step 3. In step number 3, the activator protein now attracts a histone kinase, HK. So we were at this step. Now a histone kinase comes in. So you can see the activator protein here, still bound to the histone acetyltransferase, but now also attracts this histone kinase. You can have step number 4, where the histone kinase phosphorylates serine 10 of histone H3. And again, you cannot skip steps. This can only occur after acetylation of lysine 9. So with the histone kinase here, notice the symbol again, histone 3, serine 10, H3S10. You get the phosphate group here. You get the phosphate group on the other one. You're done step 4, you can go to step 5. Serine modification signals the acetyltransferase to acetylate lysine 14 of H3, of histone H3. So after you add the phosphate groups, now the acetyltransferase can add on the acetyl group to H3K14 and the other H3K14. Now, the histone code for transcription initiation is written. You have modified the tails. You are done writing the code. So if you're done writing the code, well, there has to be a code reader. And the code reader in this case is actually TF2D and a chromatin remodeling complex. They bind to the acetylated histone tails and initiate transcription. So TF2D, you can see right here, binding to the tails, chromatin remodeling complex binding to the tails, and they're going to work together. They're going to initiate transcription. You may have to move the histones, you may have to transcribe after it, but definitely you're going to start initiating transcription here. Now the processes here were a little bit complicated. Make sure you review, make sure you go back to take a look at it, particularly before you look at my next comment. My next comment is essentially, look at the end here. At the end of all these histone modifying steps, the readers come in, and one of the things that comes in is this chromatin remodeling complex. So if I flip back to this slide where we showed you the 4 major ways activator proteins can alter chromatin, we said we're talking about number 4 here with these histone modifying enzymes. Well, notice at the end of our example, we called in a chromatin remodeling complex. So we have here the 4 major ways, but they're not completely independent. They can definitely overlap, right? At the end of our interferon gene promoter example, after the histone modifying enzymes, we then went through chromatin remodeling. So that can also, say, include histone chaperones. You can have a lot of extensive overlap. And that's really how your various activator proteins work. In the last little bit for this lecture, we want to talk about transcriptional repression. Unlike prokaryotes, eukaryotic repressor proteins rarely compete with RNA polymerase for access to DNA. Instead, they use a variety of mechanisms to inhibit transcription. One method is that they can interfere with activator function. Say we have our DNA region. Imagine this is a data box as an example. Further back, you might have a binding site for an activator that overlaps with a binding site for a repressor. So if I look at my activator protein, drawn for you quite well, has a DNA binding domain, has an activation domain. Well, if you look at your repressor protein, the repressor protein has a DNA binding domain, and up here, it's going to have a repression domain. And so if you say make more of the repressor protein, or maybe it binds to DNA a bit better, well, it's going to outcompete the activator protein. And instead of the activator protein here, you'd have the repressor protein. So you can have competitive DNA binding. Another mechanism is that you can have masking the activation surface. So again, you have your DNA here. You have the binding site for the repressor, binding site for the activator. Activator protein has a DNA binding domain and an activation domain. For your repressor protein, you have a DNA binding domain. You have a repression domain. Notice the repression domain binds to the activation domain and stops it from working. You can also have direct interaction with general transcription factors. So maybe you still get that DNA looping, right? DNA is not broken, just really long. Further back, you have the binding site for the repressor, and the repression domain here stops the general transcription factor. And maybe this is at a better position than the activator protein. Really depends, but that's an option. You also have transcriptional repression by altering chromatin structure. You can actually recruit chromatin remodeling complexes. In this particular case, though, the chromatin remodeling complexes, It's going to mediate repression, right? So you're going to make it even more compact. So again, here is your repressor protein, nice DNA binding domain. It's got the repression domain, calls in a chromatin remodeling complex. You can envision ATP would be used here too. And now you're going to roll it, but you're going to roll it closer together, make it even more compact. You can also have transcriptional repression by altering the histone code. You can do that. You can have the recruitment of histone deacetylases. Again, repressor protein with a repression domain calls in a histone deacetylase, removes the acetyl groups from the histone tails, right? So they don't show you the tails here, but you'd of course be modifying the tails, as in removing acetyl groups from the tails. In the last mechanism, you can say recruit a histone methyltransferase. Again, repressor protein calls in a histone methyltransferase. And again, you modify the histone tails, not drawn for you perfectly here, right? The methyl groups in blue kind of look like they're modifying the DNA, but not quite, right? You want to modify the histone tails. After you modify the histone tails, you can have these green proteins bind. These proteins might bind to the methylated histones. Notice now there's more proteins surrounding this area. It's even harder to access the DNA here. Now I want to quickly mention the histone readers and histone writers when it comes to eukaryotic transcriptional repression. Guided by gene regulatory proteins, these histone readers and writers can establish a repressive form of chromatin. So we'll show you this figure over the next couple of slides, but as a very quick overview, read again your DNA around these histone core proteins. Here's your gene regulatory protein. So this is going to be a repressor, a transcriptional repressor protein. It's going to have a DNA binding domain, a repression domain. In this case, you call in a histone modifying enzyme, a writer. So this is going to modify the histone tail. Again, it's shown for you in the core here, but it's going to be in the histone tail. Now, what happens is a reader is going to read that. So here is the code reader protein. And this code reader protein actually calls in a writer to write on the next histone tail, the next histone core. And that keeps on going. A reader reads that, calls in a writer in blue for the next one. And it keeps on spreading, keeps on spreading. Notice the reader here, complex sometimes with the writer, complex sometimes not with the writer. But essentially what happens is these proteins start to compact the area, right? It's going to make it pretty condensed. So we're going to see that this histone code can spread, and this chromatin gets pretty stabilized. Spreading of the histone code along the chromatin can be carried out by reader-writer complexes. So up close, again, we have our gene regulatory protein. It's going to call in this particular case, a histone modifying enzyme, a writer, and you're going to write on the tail. And what's going to happen is a reader will read that code, call in a writer to write on the next tail, and that keeps on going. And these reader-writer complexes really start to spread the histone code. Notice the reader, sometimes with the writer, sometimes without, depends on the specific case, really starts to compact this particular region. Now, there's actually additional steps shown for you here, because what happens next is this DNA methylase enzyme comes in. As it says, the DNA methylase enzyme is attracted by the reader and methylates nearby cytosines in the DNA. So we're no longer talking about histone tail modification only. Notice in this particular case, this DNA methylase enzyme directly modifies the DNA. Instead of just cytosine now, it's a methyl cytosine. What then happens is DNA methyl binding proteins bind to the methyl groups and stabilize the structure. So here are DNA methyl binding proteins. They bind to the methylate cytosines. Take a look at this DNA now. It's really compact. There's proteins on all the different sides. It's going to be really tough to get access to this information. It's going to be really tough to get any transcription here. So the DNA here is really condensed. It's really repressed. Transcription is really repressed. Methylation and therefore gene expression patterns can be inherited in a process we call epigenetic inheritance. Think about what's happened here. You've directly methylated the DNA. So when, say, this cell goes to divide, well, at least one of the two strands is going to have a methyl group all along this particular region. So you know what happens in the daughter cells in the daughter cells, well, that region will also be repressed. You'll also have these DNA methyl binding proteins. And of course, if you see methyl groups on one strand, there's going to be enzymes that will add the methyl groups to the other strand as well. So essentially, these methyl groups can be inherited through standard semi-conservative replication. The daughter cells then will methylate the other strand, and it keeps on going, keeps on going. You always kind of have this particular methylation. And the methylation doesn't have to occur in the daughter cells. It could happen in the parental cell, right? Right after DNA replication, you can methylate the other strand too. So if you think about it, this is actually another form of cell memory in that after you've methylated a region, the cell is going to keep it methylated. It's always going to remember to methylate that region. Just to also mention it, because it's asked quite often, is there going to be other sequences in other proteins that stop this condensation from spreading over everywhere, right? So of course there's going to be certain genes, maybe way after here, maybe way before here, that you don't want repressed. Well, there's sequences, proteins, it'll stop that particular histone code from spreading too much. And that's essentially a little bit about transcriptional repression.